All right, so today we will continue to discuss gene expression, the second stage of the gene expression. So last week we discussed the first step of the gene expression, which would be the transcription. So the transcription, basically, we just transcribe, we convert those DNA sequence into the messenger RNA. And now the next step of the gene expression would be the translation. So the translation basically to translate those messenger RNA into the amino acid. All right. So that's the translation. And then before we start the translation, the process before we discuss the process of translation, and first of all, then let's just go through some overview of the amino acid. All right. So basically, the structure and the property of amino acid will be the first topic of this section and this chapter. So for the amino acid, we all know that the amino acid are organic molecules, and then this is the basic structure of the amino acid. In the center, we have this alpha carbon. All right. The alpha carbon, basically, you have the amino group attached to this alpha carbon, and also we have the carboxyl group attached to the alpha carbon. Okay, and then we have hydrogen ion. So these three parts are always the same for all the amino acid. The difference is only this R group. Okay, the amino acid differ in their properties due to the different side chain, which is this one, the R group. Different R group will give you a different amino acid, and then they'll give you a different property. And since this the amino acid, all right, so basically the organic molecule, they all have the special property we call it the optical activity, which is the enantiomer molecule. So that because they have the ability to rotate on the plane, so that you can have you can see different image. All right, different image. So if we basically for the amino acid, normally there are two different forms of the amino acid. Okay, we have this L form amino acid, and also D amino acid. So this is left and this is right. So basically that will be the amino group on the left of the alpha carbon, on the right of the alpha carbon. That's the left and right. Okay, and the amino acids that occur in the protein are all this L form amino acid. All right, they are all left side, the left hand did the L amino acid. So basically the amino group on the left of the alpha carbon. Okay, and of course there are some D form amino acid, but mainly they are found mostly in the bacteria. Okay, in the bacteria cell wall, you will be able to see the D form amino acid. Otherwise, in nature, those proteins, they all have L form amino acid. Then for the amino acid, normally there are 20 amino acid that we can find in the protein, right? The most popular one, right? The 20 amino acid. And based on their property, like those 20 amino acids can be classified into these 4 different groups. So one would be non-polar, which would be the hydrophobic amino acid, and second group would be the neutral, uncharged. So basically these 3 groups are polar, right? Only the first group is non-polar. These 3 are polar amino acid, and then within this polar amino acid, we have neutral, which is uncharged, and then we have acidic, which is negative charge, and basic amino acid, which is positive charge. All right. So non-polar, that will be hydrophobic amino acid, and then these 3 are polar. So we have uncharged, negative charge, positive charge. Okay, these 4 different groups of the amino acid. And then here is just the table of those 20 amino acids, the 20 the commonly seen amino acid in the protein, right? And then from these amino acids, so the first column here, that's the full name with alanine, arginine, asparagine, aspartic acid, and then second column will be the 3-letter code. Like alanine will be ALA, arginine will be ARG, okay? And then this is 1-letter code. A will be alanine, R will be arginine, N will be asparagine. Okay, and from these 20 amino acids, you will see that highlight. There are 8. 1, 2, 3, 4, 5, 6, 7, 8. All right. So the isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and the valine. All right. So these 8 amino acids are the essential amino acids. So what does that mean, essential amino acid? That means they are not synthesized in the body, and it has to be supplied in the diet. Okay, so that's why it's called essential amino acid. And then you can see even in the pharmacy, you can find these individual amino acid for sale. So you can find like lysine or methionine in the pharmacy that you can buy, and then you can take those individual amino acids, because our body, they will not synthesize these 8 amino acids. Okay, so that's why it's called essential amino acid. And those non-essential amino acids, they can be synthesized from the various metabolites, all right? And then also you can see there are some amino acids labeled as the conditional essential, like arginine, glutamine, histidine, all right? So these are conditionally essential. So that means these amino acids, although our body can synthesize these amino acids, but it may require supplementation during the certain physiological condition, like a pregnancy, okay? So that's why it's called conditional essential amino acid. Okay, so now let's discuss each individual amino acid in detail, all right? So the first group is nonpolar side chain, okay, nonpolar amino acid, and then from these 9 amino acids, okay, you can see we have 2 boxes. One box is labeled as purple, all right? So the purple here, that's the basic structure of the amino acid. And then the yellow, that will be the side chain, the R group. And then you can see the basic structure are all the same for these 9 amino acids, all right? And only the side chain are different, all right? Only the side chain are different, okay? So the non-polar amino acid, they have the non-polar side chain, okay? And they are critical, important, right, in the for the process, okay, to drive the protein to fall into the 3-dimensional structure. As a matter of fact, hydrophobic interaction, which are the interactions between these nonpolar amino acid, would be one important interaction to maintain the protein 3-dimensional structure, okay? And glycine. So you can see the glycine, all right? Glycine is only 1 hydrogen here, all right? And the glycine is the smallest amino acid, and has 1 hydrogen ion, the atom, as the site, the R group. And it is the only amino acid that is not optically active, because you will not be able to see the different form of the glycine. All the rest, you can have these different form, all right? So this is glycine, all right? Glycine, and because the glycine has a small R group, okay, small R group. So it provides a minimum of a stereo hindrance to the rotation about the bonds when you connect the glycine in the peptide, all right? So they provide a minimum of a stereo hindrance to rotation. And therefore, glycine can fit into a very crowded region of many peptide chain, okay? And so one of the very commonly you heard a lot, the term collagen, okay? Collagen basically is a rotationally restricted fibrous protein. And the collagen has the glycyl residue, okay, in about every third position in its polypeptide chain, okay? So glycine, you can see glycine in the collagen, all right? So that's glycine, and also glycine and the taurine, they are conjugated with the bile acid, okay? That's the bile acid, that's a product derived from the cholesterol before they are excreted into the biliary system, okay? The conjugated bile acids are amphipathic and are important in the lipid absorption, okay? And also glycine is a neurotransmitter. It's an inhibitory in the spinal cord and the excitoria in the cerebral cortex, okay, of the forebrain. That's a glycine, okay? It's glycine. And the proline. Here you see the proline, right? The proline has a benzene ring. This is benzene ring, and the benzene ring basically you have this nitrogen here. Okay, the amino group, the nitrogen, all right, is bounded to a 2-carbon ion, all right. C atom, the nitrogen here, the amino group here, bound to this 1 and 2 carbon atom, all right. Therefore, the amino group of the proline is a secondary amine, and then the proline is often called an amino acid, all right, amino acid, because this nitrogen atom is bounded by these 2 carbon atom, okay. So the amino group of the proline is a secondary amine, and so we call this amino acid. And then phenylalanine, all right, see the phenylalanine and the tryptophan, all right, see this ring here, aromatic ring, all right. So phenylalanine and the tryptophan, they both have the aromatic ring. And this ring, okay, also contribute to the, when you try to measure the protein concentration. And this aromatic ring contribute to the process that when we try to determine the amount of protein in the solution, which we will perform the experiment when we meet in person next month, all right? We will do the protein concentration determination. So remember these 2, phenylalanine, tryptophan, all right? They have this aromatic ring, which is important when you try to determine the protein concentration. Another one would be this tyrosine here. This tyrosine also has the aromatic ring. Okay, now we'll discuss later. And then the side chain of methionine, we have a sulfur atom in this hydrocarbon group. Okay, so that's the methionine. And then you will find that this is one that contains the sulfur, and the other one will be the cysteine, also contain the sulfur. These are the 2 amino acids that contain the sulfur. Okay, so this is basically the nonpolar side chain hydrophobic amino acid. And then the neutral but the uncharged but the polar amino acid. Okay, so these 6 amino acids are polar side chain. Okay, and they're basically the side chain are uncharged at the neutral pH. Okay, and the R group of these polar side chain amino acid, they can form the hydrogen bond with water, and then play a variety of the nucleophilic role in enzyme reactions. And these amino acids are normally stable in water than a non-polar amino acid. And as I said, the tyrosine here, so the tyrosine, you have this hydroxy group, and then it's bound to this aromatic ring, aromatic hydrocarbon, and then you have aromatic. This contribute to the protein concentration when we try to determine. And then, and then you can see the side chain, basically in the side chain, you can see the hydroxy group, hydroxy group, hydroxy group, right? And the hydroxy group in the cysteine here contain the CO group, right? So we have the CO instead of the hydroxy. This is called CO, and this one can form the disulfide bond with another cysteine, which is also one of the important interaction to stabilize the tertiary protein tertiary structure. Okay. And then the other two group here, right? The other two group here, we have the acidic amino acid. Basically, you have negative charge, right? So you can see the carboxyl group here, right? Carboxyl group, because they lose a proton. They lose a proton, so that's why they become negative. And because they are the proton donor, all right? Aspartic acid and glutamic acid, because they are the proton donor, they lose the hydrogen. They are the proton donor, so they are acidic, okay? Acidic amino acid. And since they lose the proton, so they become negative, all right? And then for these three, all right, the basic amino acid, all right? And so they are the positive charge. Okay, see that they are proton acceptor, so they are basic. Okay, and they have the positive charge. So that'll be lysine, arginine, and histidine. Right, these three are positive charged amino acid, and these two are acidic amino acid. Okay. And then from this side chain that you can see, so you can see this for the non-polar, they basically have either hydrogen or methyl group, all right, or the aromatic group in the end. So that's why it's a non-polar. But for the polar, you can see you hydroxy, and the CO group, hydroxide, and oxygen here. They can form the hydrogen bond, right? They are polar side chain. No, non-charged, uncharged amino acid. And then this carboxyl group, they can donate their proton, lose the hydrogen ion, so negative charge, acidic. And these, the proton acceptor, so they're basic and the positive charge. Okay, so these are the 20 commonly seen amino acid in the protein. And then for the amino acid, okay, we can form the polymer, just like the nucleic acid to form the polymer, right? So they can polymerize to form the peptide and protein. And the reason they can form the polymer is because the amino group, right, and carboxyl group. For the protein, normally you put the N-terminal on the left side and then C-terminal on the right side. So the N-terminal, that will be the amino group. On the right side, that's the carboxyl group, okay? And then it's N-terminal, C-terminal. And then the polymerization, basically, it's a dehydration reaction, okay? So from this carboxyl group, they lose, carboxyl group lose one oxygen, and the amino group lose the two hydrogen, so they lose one molecule of water, and then this carbon atom form another bond with the nitrogen atom, all right? And we call this as peptide bond, all right? Peptide bond. In the meantime, you lose one molecule of water, which is here. Okay, one molecule of water. So this is how they form the polymer. And the returning ion, so basically amino acids are the ampholytes because they contain both acidic and basic group. In the free amino acid, they normally exist as a neutral returning ion, okay, which means they contain both positive charge and negative charge, right? The positive charge amino group and negative charge carboxylic group, all right, at the neutral pH. At this point, at this pH, the net charge is 0 because positive 1, negative 1, so the net charge is 0, all right? And the 0, so that means they do not migrate in the electric field at that specific pH, at the neutral pH, okay? And then in an acidic solution, right, normally below pH 2, right, the predominant species, okay, the predominant, because in the predominant species, okay, of the amino acid is positive charge, all right, in the acidic environment. So that will go this direction, all right, go this direction. So acidic environment, so it can provide the proton to the carboxyl group, all right. When the pH go down below 2 acidic condition, then this carboxyl group will accept one hydrogen ion, then the net charge of positive 1. Okay, in a basic solution, all right, normally above pH 9.7, the predominant species of amino acid would be negative charge, right? Negative charge, because you will lose another hydrogen ion, right? So here, from the amino group, you lose the one hydrogen ion, then become like this, and then there'll be negative charge, all right? So go from the neutral to the acidic, positive charge, to the basic condition will be negative charge, okay? And then the isoelectric point, which is a pi, we call it pi, the isoelectric point of an amino acid, would be the pH at which the molecule has a net charge of 0, and therefore that molecule will not migrate in an electric field, right? So the isoelectric point of an amino acid will be the pH, the neutral pH, when you have the net charge 0. Okay, let's return ion, and let's watch this video about this amino acid structure.
Although we usually represent an amino acid as a compound with an NH2 and a COOH group, this is not strictly accurate. Amino and carboxyl groups can react with each other, owing to their opposite.
So it's acidic because it's a proton donor, right? Release the hydrogen ion, right? And then here you accept the hydrogen ion, so it's a base, right?
Acid-base properties. The amine group acts as a base and accepts a hydrogen ion released by the acid's carboxyl group. The reaction product is an internal salt, in which a positively charged ammonium cation is neutralized by a negatively charged carboxylate anion. Such a structure is referred to as a zwitterion, from the German Zwitter, meaning hybrid. Note that zwitterions are highly polar, but they are electrically neutral and have no net charge.
No net charge, okay, with strong ion, okay? So from here, we have isoelectric with strong ion, right? The net charge is 0. And then if you go down the acidic environment, all right, environment, you accept one hydrogen ion, okay? And then it becomes positive charge, all right? So that's a cation ionic form. And then if you go up to the basic environment, all right, you lose this one hydrogen from here, so it becomes NH2, and that'll be negative charge, is an ionic form, okay? So that's ionization of amino acid. And here, just to show you that at the physiological pH, right, the monoamino and the monocarboxylic amino acid, like a glycine and alanine, they exist as return ions. So that means at pH of 6.9 to 7.4, the alpha carboxyl group is dissociated to yield a negative charge negative charge carboxyl ion. And then the alpha amino group, which is here, amino group, is protonated to yield an ammonium group, NH3+. So this will be at the neutral pH, all right? And then the PI, basically that will be the PK1 plus the average of PK1 plus PK2. What that means, the dissociate of these hydrogen ion accept the hydrogen ion, those PK1, PK2, and then that give you the PI, all right? And then if you go down, all right, when you go down the pH, all right, go down the pH, then the predominant molecule, right? The predominant molecule will be the positive charge because the CO receive the hydrogen ion, right? So that'll be positive charge COOH. So you increase the amount of the COOH here, right? When you reach to the 1-to-1 ratio, that will be the PK1 here. That's PK1, all right? That's a PK1 when you reach to the 1-to-1 ratio. And then again, if you go to the basic environment, so that means the predominant in the basic environment will be negative charge, right? Negative charge because the NH3 plus will lose the one hydrogen ion. Okay, so when it reach to 1-to-1 ratio, that'll be a PK2, right? So that PK1 plus PK2 and divided by 2, that give you the isoelectric point. That's a neutral pH, all right? And this figure just to show you the titration of a glycine, okay? The titration of glycine. When you begin from the glycine hydrochloride, which is this one, the glycine hydrochloride, and then you start titrate the glycine, right? And then so it goes through this PK1, PK2, and then the PI at the neutral pH where you have all the amino acid exist at this form. Okay. So that's the PI, and then the isoelectric point. Okay. So that's a pH which the majority of molecule of a compound in solution have no net charge. Okay, so you have PK1 plus PK2 divided by 2. So the example for the PI for the glycine, so the PK, the carboxyl group, amino group, all right? And so it's 2.35 plus 9.78 divided by 2, 606, right? See this here, there's a PK1 and PK2. When they reach the point that they have this 1-to-1 ratio, okay? And this is just the same as the first lab when we try to learn this the weak acid dissociate, then you have the conjugate base, right? And the same principle applied to here. And this is just a group the PK1, PK2 value of each amino acid. All right, so the PI for the glycine, that'll be this plus this divided by 2, give you 597. Alanine, 1 plus 2 divided by 2, give you 6.0. And then for this positive charge, glycine, arginine, histidine, positive charged amino acid, all right? So basically for the PI, all right, for the PI, you have to take these two numbers, all right? PK2 and PK3 and divide it by 2. Okay, remember the first one, this is carboxyl group dissociation, and this is amino group. All right, and these are positive charge. Okay, positive charge amino acid. Okay, so you would take these two, right, pK2 and pK3, and divide it by 2. So for example, lysine A95 plus 1053 divided by 2 is 974. And then for these two, okay, negative charge amino acid, all right, you would take the pK1 and pK3. Okay, so that'll be aspartic acid is 1.88 plus 365 divided by 2 is 277. All right, and glutamic acid 219 plus 425 divided by 2 is 322. So that's how you calculate the PI and the isoelectric point. Okay, and the protein, okay, that so basically that consists of a single polypeptide chain are generally considered the 3 levels of organization. We have the primary structure, secondary structure, and tertiary structure. Okay, and for those protein structure depend on the composition of amino acid and also the location of that specific region in the cell. For example, here we have a cell membrane, and then we have the peptide chain, and normally for these, so we have normally the hydrophobic amino acid like to bury inside, for example, in this case, inside the membrane, right? And then hydrophilic will be exposed outside, okay? So that's the amino acid. And the amino acid content, they can determine the protein structure and function. And there are 3 levels of the structure, that's primary, secondary, tertiary for single polypeptide. For the protein that contain 2 or more peptide, then we will discuss another level of the structure. That will be the quaternary structure. Okay, so for single polypeptide chain, you have the primary structure, secondary structure, and the tertiary structure. For the peptide, for the protein contain 2 or more peptide, then that will be the quaternary structure. Okay, so the primary structure basically will be the amino acid sequence, which will be the sequence of amino acid in the protein, determine the nature and the activity of that protein. And the primary structure can be determined directly by the DNA sequence, all right? Because later on we will learn the genetic code. So the basically the genetic code, they will translate those DNA sequence to the amino acid, all right? And then those DNA sequence translate into the amino acid sequence, and that's amino acid sequence that will give you the primary structure. Okay, and the well-known example of the primary structure that can influence the cell function would be this hemoglobin in the sickle cell anemia, because one amino acid change that will change the protein function. Okay, that's a primary structure, as you can see here, from N-terminal amino group, from the N-terminal, and then to the C-terminal carboxyl group. And this will be just like a DNA. You have 5' N, and then this will be like a 3' N. Okay, and the sequence of amino acids. So these will be the primary structure. And the primary structure of the amino acid. Okay, so here just to show you the peptide bond. Okay, how they form the peptide bond, and then so from here, this is alpha carbon, right? Amino group, carboxyl group, and then they form this peptide bond with the next amino acid. Okay, so the amino group, the peptide bond here, and then another peptide bond here, and then you can see the side chain. See, one will be on in the bottom, the other one will be on the top, and bottom and top. So basically, the most peptide bond exists in the trans configuration to keep the side chain as far apart as possible. All right, because they try to keep this side chain, as far apart as possible. So that's why they have to put this one down, one up, down, up. So that's called trans. If it's all in the same side, that'll be cis. All right, that would be very rare because normally the peptide bond, they will try to arrange the side chain in the trans configuration. Okay. And then, so in almost all the protein, the hydrogen bond that make up the secondary structure involve the amide proton of one peptide group and the carbonic oxygen of another peptide group. Okay, so that will give you this secondary structure. So for example, this the hydrogen bond, okay, the hydrogen bond, the oxygen, the carbonic oxygen, all right, and then we have this hydrogen, all right, from the amide group here, all right. So there's a peptide bond, another peptide bond. See the number 1, number 4, and then you skip 2, right? And then you skip another 2 with 7, and then you skip 2 will be 10. So basically, this oxygen, all right, from the peptide bond will form the hydrogen bond with this hydrogen ion in the next amide group. Okay, so that's how they form the peptide, the hydrogen bond. Okay, they form the hydrogen bond, and then the coil. See, like this coil, and they folded, right? That's a secondary structure, and they form this secondary structure basically rely on this hydrogen bond, all right? And this will be the alpha helix coil, the very commonly seen secondary structure. Okay, alpha helix, and then you can see the hydrogen bond between this oxygen from this carbonate here, oxygen form the hydrogen bond with this hydrogen ion here from the amide group here. Okay, so every four amino acid, they will form this hydrogen bond, hydrogen bond, hydrogen bond. And here, just to show you the hydrogen bond here, this NH and the CO, right? NHCO, okay? They form this hydrogen bond so that they can have this alpha helix structure. And then the beta sheet, okay, beta sheet, basically the peptide backbone, the beta pleated sheet is almost completely extended, just like this, extended, all right? And then they form this hydrogen bond, okay? The hydrogen bond within the peptide bond, okay? And then if you see this parallel sheet are typically a large structure. So basically, they have more than 5 strands. That's the parallel structure. For the anti-parallel, you see from the N terminal to C terminal, this direction, N terminal to C terminal, this direction, these anti-parallel sheets, okay, normally they can consist as few as only 2 strands, okay. And this is detail. You can see the hydrogen bond between these 2 strands, okay. This, that's a parallel. And you can see this amino group and then carboxyl group here, and then amino group, so this anti-parallel. And then they all form this hydrogen bond between this hydrogen ion from this amide group and the oxygen from the carbonic group. Okay, you see this hydrogen bond. Okay, these are the force to maintain the secondary structure. Okay, so for the primary structure, we have the peptide bond to maintain the primary structure. For the secondary structure, then we need the hydrogen bond. The hydrogen bond basically between the amide proton, all right, this amide proton, all right, and the carbonyl oxygen, all right, carbonyl oxygen and the amide proton. So they form this hydrogen bond, okay, to stabilize the secondary structure. So this is just another figure to show this, the oxygen, hydrogen, oxygen, hydrogen, okay, to form this secondary structure. And for the secondary structures, several important role in the cell, because a lot of the transcription factor, okay, transcription factor, they need to bind to the DNA, all right? They need to bind to the DNA. Therefore, their secondary structure play an important role to help the transcription factor to bind to the DNA. And there are several kinds of the secondary structure. So for example, we have the zinc finger motif, right? Zinc finger motif are the domain that frequently found in protein that bind to the DNA. Okay, they have a two beta sheet, like a two beta sheets, all right? And follow, you see the two beta sheet, and then followed by the alpha helix here, okay? The zinc finger domain, especially in those transcription activator. And also the leucine zipper, okay? Leucine zipper also found in the transcription factor, okay? And this is the helix loop helix structure. Is also a transcription. This is a helix turn helix motif. The also transcription factor can bind to the DNA. So now from the secondary structure, we have alpha helix beta pleated sheet. Then they can form this tertiary structure in a polypeptide, in a peptide chain. All right, because a long peptide, you have several alpha helix and beta pleated sheet, and from those second structure, then they form this tertiary structure. All right, and quaternary structure, as I say, the quaternary structure when you have two or more peptide bond and the peptide chain, they form this quaternary structure. Okay, and then for the tertiary structure, basically it's a three-dimensional arrangement of all the atoms in a single polypeptide. Okay, the interaction between the side chain play an important role in the folding of the protein, and the side chain including the peptide backbone hydrogen bonding, the peptide backbone hydrogen bonding. That would be the secondary structure that we see. All right, like alpha helix, beta pleated sheet. All right, we have the oxygen from carbonyl group and the hydrogen from amide group. They form the hydrogen bond. Okay, so that will contribute to the tertiary structure. And also on top of that, we have the hydrogen bond between the polar amino acid. Okay, polar uncharged amino acid. They contribute a hydrogen bond, right? And then we also have the hydrophobic interaction between the non-polar amino acid. Then we have this electrostatic interaction between the oppositely charged amino acid, and then disulfur bond between the cysteine. So all these forces contribute to the tertiary structure, okay? And then they contribute to the tertiary structure, which will be the three-dimensional structure, and of course the protein. Okay, because those are variation of the sequence and also the property of different side chain, they can form a different kind of the tertiary structure. But all the protein, they only have one functional conformation, and that conformation, the only conformation that make this protein functional, okay, as a native structure. And so if you want to denature the proteins, which means you break those interaction and those forces that contribute to tertiary structure. And if you break those interactions, then the protein will no longer be functional. Okay, they lose their function. Okay, that's the denaturation. And then what forces can break those interactions? So we have a heat, all right? For example, heat and also some chemicals. So they can break these interaction. The interaction that I just mentioned. Okay, the hydrogen bond from the peptide backbone, the hydrogen bond between the polar uncharged amino acid, the hydrophobic interactions between nonpolar amino acid, electrostatic interaction between oppositely charged group, and the disulfide bond. So they can, if you break all these bonds, then the protein is denatured. Okay, but they still maintain the primary structure. Okay, they still maintain the primary structure. Okay, so this is just the detail to show those interaction of forces contribute to the tertiary structure. So we have this hydrogen bonding. You see the hydrogen bond between the polar uncharged amino acid, right? And then we're hydrophobic between those non-polar amino acid. And then disulfide bond between the cysteine, the electrostatic interaction between this is that produce this ionic bond between the negative charge and positive charge amino acid. Okay. These stabilize the tertiary structure. Okay. And then we know that many protein that consists of several individual polypeptide chain. Okay, especially for those protein, their total molecular weight is much greater than 50,000 Dalton. All right, and the assembly of these multiple subunits now give us the quaternary structure, and the quaternary structure is the final level of a protein structure. Okay. That's the quaternary structure. So all the forces contribute to the tertiary structure, also contribute to the quaternary structure. And for those, as I mentioned, the quaternary structure, you need to have 2 or more polypeptide, so that when we have a monomer, okay, that's one single peptide, and then we have 2 identical polypeptide. We form the, if you have 2, then they can form a dimer. If you have 3, then you can form the trimer, and 4 you can form the tetramer. Okay, and sometimes the protein need to have other component to join, and so that the protein will become functional. And those protein are the conjugated protein if they have the components other than the amino acid. All right, and then the non-protein component of a conjugated protein would be the non-protein prosthetic group. Okay, so lipoprotein, so that'll be lipid plus protein, and lipid is the prosthetic group, and glycoprotein will be carbohydrate plus protein, and metalloprotein, the metal ion plus the protein. Okay, that's the quaternary structure. And here's hemoglobin. So you can see we have this alpha subunit, we have 2 alpha subunit and 2 beta subunit, and they bind this iron. Okay, this is hemoglobin. And then the next here, just to show you the single amino acid substitution in the protein causes the sickle cell disease, and that's the number 6 amino acid. If you change this from glutamine to the valine, right, then this is when they produce this each subunit, the peptide, and then the quaternary structure. All right, so this would be the functional quaternary structure, so they can bind the oxygen, okay, without any problem. But if you have the sickle cell, basically they change the conformation, then this whole protein is no longer functional, and they cannot bind, they cannot carry the oxygen efficiently. Okay. that will give you the sickle cell hemoglobin. Okay, so that basically we just discussed from the amino acid and then also the protein structure. And now we just want to discuss the protein structure a little bit further. Okay, basically we know the protein can be grouped into 3 large classes based on their structure and solubility. Like we have fibrous protein, globular protein, and the membrane protein, right? And of course, the protein, they have to fold into a functional protein. So that means they need to have a specific conformation, right? And this can be done by a process, okay, that helped by the chaperoning, right? The chaperone basically will receive the newly synthesized peptide and then fold it into the functional conformation and release it, and so that'll be the functional protein, okay? And then for the fibrous protein, they contain the polypeptide chain. They organize approximately like a parallel along a single axis like this, all right? They produce a long fiber or large sheets, and these protein tend to be mechanically strong and resistant to solubilization in water. And so the fibrous protein often play a structural role in nature. Okay, that's the fibrous protein, like a collagen. All right, collagen. Okay, that's an example of the fibrous protein. And the globular protein, that's because they are like this. Okay, that's their shape is a spherical shape, so that's why it's called globular protein. It's the most abundant protein in nature, and the globular protein exists in an enormous variety of 3-dimensional structure. And nearly all the globular protein contain substantial number of alpha helix, and the beta sheet fall into a compact structure, and that is stabilized by the polar and non-polar interaction. And the globular protein are usually very soluble in aqueous solution. Okay. The globular protein, they have many functions in the cell, okay, like binding, catalysis, regulation, transport, immunity, cellular signaling, all right? So those functions, that's globular protein. And then another type is called membrane protein. We can find the membrane protein in association with the various membrane system of the cell, okay? And for the interaction with the nonpolar phase within the membrane protein have a hydrophobic amino acid side chain oriented outward, okay? And membrane protein have a variety of function, okay, like a transporter protein, help move the substance in and out of the cell, receptor protein, okay, that's a membrane protein that is responsible for the transfer of an extracellular signal, okay? So it's just like the receptor protein on the cell surface. So the most famous one would be like right now, the SARS-CoV-2 virus, okay? The spike protein interact with the ACE receptor on the cell surface, all right? So that they can fusion into the human cell, okay? That's one of the examples of a membrane protein on the surface of a membrane, okay? Act as a receptor, ACE receptor. So the protein structure, this is functional protein structure, right? Only one is functional, and then if you disrupt this conformation, that will become a denaturation, all right, and then become inactive. So there'll be active protein, inactive protein. Okay, as I mentioned previously, all the factors that can break the hydrogen bond, disulfur bond, hydrophobic interactions, and also ionic bond will be able to break the protein structure. So in the few slides earlier, we mentioned the heat. When you increase the temperature, that will break the protein structure. And chemical agents like organic solvent, detergents, okay, especially the denaturing solute, they can denature the protein. So like the SDS, the protein SDS, okay, they can disrupt the hydrophobic interaction and the electrostatic ionic interaction as SDS, okay. SDS can disrupt the hydrophobic interaction and the ionic interaction. And then another detergent, urea, and the guanidine hydrochloride, okay, they can form the hydrogen bond with the protein that are stronger than those within the protein itself. So they can disrupt the hydrophobic interaction, okay. The guanidine hydrochloride, okay, they can disrupt the hydrophobic interaction like the SDS. And the reducing agent like a beta-mercaptoethanol, right? The reducing agent DDT, beta-mercaptoethanol, you can reduce the disulfur bond bridge between the two cysteine. Okay, so all these are the factors that can disrupt and denature the protein. All right, and to determine the protein structure, basically nowadays they have two popular techniques people can use to determine the protein structure. One is the X-ray crystallography, right? This is X-ray crystallography. And the other method will be the nuclear magnetic resonance, MMR spectroscopy, okay? And then for the X-ray crystallography, it require the protein crystallization, which means you need to for this X-ray crystallography, so that means you have to use the genetic manipulation to clone the protein you want to study into a bacteria cell, and then let the bacteria cell to express that specific protein, and then you crystallize the protein, right? You crystallize the protein. Like here you have crystallized the protein, a single object here, all right? Then you have these X-ray cells, okay, to go through the crystallized sample, right? So then the X-ray can be distorted, right, when they pass through this object. And then the structure of the protein within a single crystal detect the pattern, right? Detect the pattern that the X-ray is diffracted. And then from this pattern here, then you collect the information, and then you can use a computer to calculate and then to determine the protein structure. Okay, so that's the X-ray crystallography. So you need a large, well-formed crystal of highly purified protein. Okay, so that's X-ray crystallography, and then the MMR spectroscopy. Okay, that's another technique that can be used to identify the three-dimensional protein structure. So basically, in this technique, a solution of pure protein is bombarded with radio wave, okay, as it is held in a strong magnetic field. Then the radio wave causes the atomic nuclei of each molecule to spin and eventually align, and then the movement emit energy that is detected by a computer and analyzed by the Fourier transformation, and then they can determine the protein structure. That's MMR. Okay. And here just to describe all the function the protein have in the cell. Can be the enzyme, can do the transport, storage, motility, structural, defense, regulatory protein, okay, all kind of the function. That's for protein. All right, so that's the first half of the chapter 3, okay? Any question